All right, let's continue. So I'm going to come back to the STRUCTAL algorithm, which uses iteratively a dynamic programming algorithm to compute the scores of matches from distances every time iteratively. We are going to see that. But let's first take a look at how DALI works. So DALI is the acronym for distance matrix alignment. So it works on those intra-atomic distance matrices. And so the first algorithm was in actually 1993 in Journal of Molecular Biology, again by Holm and Lisa Holm and Chris Sander. And then they also, by using the method on all pairs of protein structures, they also wrote a paper in Science mapping the protein universe. So they also, after that, they also talked about how they can provide a database search for those structural similarities. So it uses intramolecular distances. Here it's called intra because the molecular distances are within the same protein structure. Intermolecular is between two different molecules. If we look at, so RMSD is like actually an intermolecular distance measure because it's the distance measure between two different protein structures. Intramolecular means that it converts the protein structure into, by looking at the distances between itself. Okay, so inside that same molecule, the distances between the same molecule. So let me represent each protein as a 2D matrix of intramolecular distance. We have protein A and protein B now represented completely as 2D matrices. And our goal will be to find sub-regions, sub-matrices here like this, sliding one matrix over another, finding local parts. At the end, our goal will be to find a similar sub-matrix in both of them. So what they do is, as I told already, they break the matrix just like the IDM BLAST. They try to find small, nice matching regions. So they first break each matrix into overlapping submatrices of fixed size, probably 8 by 8. And then pair up similar submatrices. Just like in BLAST, there was high scoring pairs. This is like those high scoring pairs. Similar submatrices between a pair, a similar submatrix contains one submatrix from protein A matched to another submatrix from protein B. So we have this match. And if we are given these pairs of matching small matrices, can we assemble them from this large set of high scoring pairs? Can you select a consistent subset? Which means that, for example, if a submatrix is matched to one here, that same submatrix or part of it cannot be matched to someplace else. So it has to be, our goal will be to consistently grow, just like extension. The third bullet here is like the extension step of the BLAST algorithm, where we try to extend the high scoring pairs into a larger alignment. So here is a distance matrix of two protein structures that are different, shown here. This is not a square matrix, as you can see, but here, in order to, I think it should be, in order to have a diagonal like this, we have to, I mean, if this diagonal it has to be a square, their size have to be matching, right? I think this is showing two matrices, two intramolecular distance matrices for one protein, and the other one here, since it's symmetric in itself, the lower triangle is now shown. Lower triangle shows the distance matrix for another one. As you can see, this one is not symmetric here because this is a different protein structure. So we are trying to find similarities from here to sub-matrices that are similar here to sub-matrices that are here. So the distance matrix is computed by just looking at all pairwise, and they also, the original algorithm, it looks at alpha carbon atoms too, carbon alpha, as you can see. and find similar submatrices. And then we call these submatrices of fixed size contact patterns. Well, they use hexapeptide contact patterns. What does this mean? It's actually the submatrix that they're looking for is not 8 by 8, but 6 by 6. So this hexapeptide tells me that hexa is the Latin for 6. So 6 by 6 submatrices are tried to be matched. And this pair list is our high scoring pair list, and then they try to, this is the assembly of these high scoring pairs into a larger alignment is a non-trivial combinatorics problem, and the same is true for that combinatorial extension algorithm that I mentioned, the other algorithm by Philip Byrne and Elias Shindiello. And we need to combine these high scoring pairs in such a way that they are consistent. And they use, for this part, they use different methods. They use Monte Carlo optimization, branch and bound, neighbor walk. So they try different methods to do this combinatorial extension. And here's just a figure that shows an overview of how it is done actually. For example, these are two protein structures. I have the contact pair. So look at these secondary structures, AB. Okay, these are like, or two parts of the protein structure. So the distance between AB is represented here as a submatrix in the first protein structure. So this purple one, the pink submatrix shows here the relationship between A and B as a submatrix. Okay, if in the other protein structure I have a similar matching pair of secondary structures, which let me call them A prime, B prime. In the second protein structure, which as you can see is larger, we will have a similar pink submatrix which matches this one. Okay, so here, which tells you that if these pairs are matching, A should be aligned to A prime and B to B prime. So this is one pair. Okay, but the relationship between this AB and A prime B prime is represented as a single 6 by 6 matrix, submatrix here. Imagine we find in another part, we find another submatrix B and C, and B prime C prime here. That part is the turquoise submatrix, shows the relationship between B and C and B prime C prime. But in reality, as you can see, since these are both anti-parallel beta strands and the distances between them are all black, probably this will also be like pinkish color if we represent the content of that hexapeptide submatrix with some color coding. This should also be like pink, right? Because it's also similar to that one, but just for the purpose of showing how that combinatorial extension is done, it shows, let's assign it a different color here, just to indicate that submatrix that is between in one protein and the other protein. Now, combining all these two together, I have one pair, the pinks and the turquoises, and here the yellow one also shows the relationship between A and C and A prime C prime, if I'm not mistaken. And the goal is just try to combine all these together in a consistent manner. So it tries to do that for all high scoring pairs, which may be just more than two. So it's the main algorithm is complex, and this is the part that I'm not going to go into detail. Just know that as invariant features, it uses the intramolecular distances, and those distance matrices are tried to be aligned in the DALI algorithm. You're not responsible from this Monte Carlo optimization part or the branch and bound method. These are the technical details that I mentioned. And from the log algorithm, you only need to know that it's a combination of secondary structure and atomic level comparison. So it uses helices and strands. They are represented using line segments as vectors and dealt with first. And then atoms are aligned later. I'm not going to go into talking about the lock algorithm in detail. You're not responsible from lock also in the final exam. So yeah, and there's also another algorithm, VAST, which is also similar alignment of secondary structure elements. Here, the quality of a structure match, as I said, we have to look at RMSD and length, but we may also compute a statistical significance similar to BLAST's E-value. Many of the algorithms like DALI, they give you as a result z-score, which tells you how different the alignment is from the null distribution of alignments between unrelated protein structures. So they're compared against a random match. And the only thing that I'm going to mention that is remaining, so lock and WAST, you're not responsible from the details, and you're not responsible from the branch and bound and Monte Carlo part of the DALI algorithm. Now I'm going to talk about in detail another algorithm, which is depicted here, the STRUCTAL algorithm. That will be the final thing that we are going to discuss in structural alignment. So the idea is really intuitive, but it's not doesn't give good results as DALI and CE. So that's therefore it's not very popular. But I just want to show you that how the methods that we have learned for sequence alignment can also be extended to work on structural alignment. So this structal uses, the idea is we first start with an arbitrary matching between the points of the two structures. Because in the second step, it superimposes the proteins to minimize RMSD. So the idea is this. We have two protein structures and we want to figure out whether they are similar in shape or not. Let me get a new slide here. So imagine I have one structure that is here in 3 dimensions, and I have another one that is here. Okay, so in order to find which parts are similar to each other, what we first do is we select their first, in order to find whether similar in shape or not, it's difficult when you just look them when they're apart, right? You need to get them close to each other. Translate and rotate this close to this one. So this is how the algorithm actually, it uses not the intramolecular distances, but to find similar shape, it uses intermolecular distances between the atoms. But initially, if your protein structure is very far away from the other, all the similarities will be very bad because the distances are large, right? We need to get them as close as possible to each other. So in order to get them as close as possible to each other, we first need to translate and rotate this with respect to some matching. So the algorithm says, why not just randomly match a portion of this? For example, let me match this portion to this portion as much as possible. So if for example, this one contains M and this contains N atoms, N amino acids, if M is larger than N, we may, so this is not W, this is N, we may randomly assign each one of these N amino acids to to N amino acids here. So some of them will be left unassigned. But so whatever random assignment we start with some random. So that's one of the disadvantages of this algorithm. Every time I run structal, the result may be different. This is non-deterministic because the first step is a random. It has some randomness. Start with arbitrary alignment. If you start with a different arbitrary alignment, the final result could be different. It may not even be optimal. Okay, so this is one disadvantage of this method. And after we have this random alignment, we superimpose them in order to have the second structure on top of the first one as much as possible. So if this was the second structure, whatever it is, so it becomes something like this. After this point, after the second step, what we do is we compute how an amino acid is between two amino acids from the first protein. Let's call the first protein A, the second protein B. We in order to determine a scoring and penalties, match scores and mismatch scores between the amino acids in protein A and amino acids in protein B. If this was sequence alignment, we would just refer to a BLAST 62, BLOSSOM 62 matrix to find whether I can match this amino acid to the an amino acid here to amino acid here, like, right? I mean, in order to do dynamic programming alignment between these two sequences, I have to have a scoring mechanism. What is my match score? What is my mismatch score? And in sequence alignment, this matches and mismatches are done by looking at their amino acid types. Now we are trying to do a structural alignment. And in that structural alignment, the scoring mechanism that you're going to form is by using the distances between an amino acid here and the amino acid in the other protein sequence. So what we do is we compute a distance. Let me erase this right hand side quickly, and then I'm going to use that part. So let me quickly erase this part. Okay, now we are going to conduct a similarity score similarity matrix, which is like this. Look at this. It's an m by n matrix, or n by n, it doesn't matter. So one of them contains m amino acids, the other is n amino acids. So this m by n matrix that I'm going to form is going to have, for example, the ith amino acid in structure B. This is structure B, this is structure A, and the jth amino acid. So, sorry, the jth amino acid is here. So the I and jth amino acid here, the score of matching I with J will be computed by using the distance between the ith amino acid and jth amino acid in these two structures. And this distance, as you can see, is between two different molecules. It's interatomic distance. I will compute this distance and convert it to some similarity value, because distance is the reverse of similarity, right? I want, if I'm going to use this entry as a similarity value, it has, the larger it is, I have to have good scores. The lower it is, I have to, it have to be negative, like a penalty. The distance is between 0 and some large number here. Let's not m, let's call it the maximum distance, d, here, between them. And the smaller the distance between ith and jth amino acid, I want to have larger score here. So what they do is they, by just some arbitrary scoring measure here, look at this highlighted part, the distance between I and J. So this is a constant, M is a constant. By using these two constant numbers, the score, the distance between I and J, since it appears in the denominator, the smaller the score, the smaller the distance, the larger the score is going to be. For example, when Dij is equal to 0, the entire denominator is going to be 1 plus 0. So this is 0 divided by some constant square, it will be 0. And 1 plus 0 is going to be 1. So m is like your largest score. The constant m is your largest score that is possible in your scoring matrix. Whatever you want that to be, you have that one. And d0 is like your largest distance, probably, in which you compare your Dij against. But so it's also a constant. But the thing is that everything is going to be converted into a similarity score, all distances. So after the superimposition between every amino acid pair between two protein structures, I compute the distance between them and I convert it to a similarity score. So I will have Sij's all over, S1, S11, S12, S13. So this S13 all the way to Smn. This is computed, these scores are computed by just looking at these pairwise distances. This matrix is not the dynamic programming partial scores table. This is our matrix that we are going to use to replace Blossom 62. Because as you can see, the scoring table tells me what is the score of aligning the I amino acid against the J amino acid. What is the score of aligning this with that? What is the penalty of aligning this with that? So this M by M matrix that I form after I do the superimposition will act as a Blossom 62 matrix. After you have the match and mismatches between all pairs of amino acids, what you can do is you can try to align A with B by using the scoring matrix. After the alignment, what is going to change? The alignment is going to tell you this amino acid matches with this amino acid now. Okay, the initial matching we had to do this superimposition will be changed after using this scoring matrix and running a dynamic programming alignment, running a DP by using this as my Blossom 62 matrix. The dynamic programming alignment will tell me with that I'm going to find a region, a sub-region between A and B, which is matching. I will match certain amino acids with respect to each other based on their closeness, and this dynamic programming alignment will provide me a new matching. This is really critical. I started with random matching of amino acids. I computed a score matrix after superimposing them. Using the scoring matrix, I run dynamic programming algorithm to get me a new matching of amino acids. With these new matching, what I can do, I can resuperimpose them because the amino acids are, instead of the random started matchings, now I have a better matching. Now let's use this matching of amino acids to do the superimposition. So we do the superimposition again. After superimposition is done, the red protein structure is going to change its orientation, its location. After it changes its rotational orientation, what does change? My scoring matrix is going to change. So this is done iteratively over and over again until the final alignment does not change. Okay, so that's basically, you may look at the final RMSD whether it's improved or not. If it doesn't improve, you stop your alignment like this. So iteratively, we first we superimpose them. After superimposition, we compute ourselves a scoring matrix which tells which amino acids are close to each other in 3D space. And by using this scoring matrix, we use the same dynamic programming alignment we did for sequences. The only difference is that instead of you may use certain gap penalty, whatever gap penalty you want to use, you may use it. But for match and mismatch scores, we refer to this table, which gives us match and mismatch scores with respect to their closeness to each other in 3D space. This will, after that, doing dynamic programming, since the scores change from the previous iteration, the dynamic programming result may change. If it changes, then superimposition is changed. So it's just like very similar to expectation maximization type of algorithms, in which you first fix something, you optimize the other. After you optimize the other, whatever you have fixed is going to change. You come back, change that, and you do it iteratively until convergence. That's what STRUCTAL algorithm does. So here, if you look at the pseudocode, we first superimpose the initial random alignment. The structural alignment matrix is what we use to replace Blossom 62. Every entry is the score for structural similarity between two pairs of amino acids. Whether it's a match score or a mismatch penalty is indicated in this assay matrix. And it's computed purely by looking at the three-dimensional distances between inter-atomic distances between two different molecules. By using this assay, we use dynamic programming to compute the next alignment, which will change the matched amino acid pairs. And here, as you can see, it says the gap cost is 0. So we can have as many gaps as possible. It uses the gap penalty of 0 here. And after that, we iterate the steps 2, 4. After we compute the new alignment, the amino acid matching pairs are changed. Therefore, we resuperimpose them considering the newly matched pairs. find the minimum RMSD, since translational rotation is of the second molecule is going to change after this superimposition again, SA matrix is going to change. So we do it, the steps 2 to 4 is iterated until the overall score or RMSD converges to a small value. And number 6 here is for non-determinism. Since the initial alignment at step 1 determines whether you're going, maybe you may stuck at a local minima depending on your initial alignment. Therefore, it's a good idea to run the algorithm a number of times by starting with different random alignments between these two structures. So this is what it says: run the same algorithm over and over with different random starts. But even if you repeat it 100 times, again, the structural algorithm overall is still non-deterministic, because it doesn't guarantee that if you run it 100 times, the best alignment you find in 100 runs, it's not guaranteed to be the same to another 100, the best alignment that you find in another 100 runs. Those are all random samples in your search space. But the idea is, like, this is a greedily walking towards a better alignment. This figure shows how it's done. So initially, alpha, beta, gamma is matched with A, B, and D. Okay, so this alpha, beta, gamma is one structure. This A, B, C, D is the other structure. Actually, there's a really good structural match between alpha, beta, gamma and A, B, E, as you can see here. Alpha, beta, gamma matches very nicely with A, B, E. They're both linear. CD should not be matched. But initially, since we don't know, we don't see this, we want to do an automated method here, we don't know alpha, beta, gamma should be matched with ABE. If it was matched with ABD initially, okay, alpha, beta, gamma matched with ABD initially in this random alignment. What we do is we compute, we try to superimpose ABD with alpha, beta, gamma so that the matched ones are as close as to each other. So the orientation of this second molecule, alpha, beta, gamma, composed of alpha, beta, as you can see, is superimposed in such a way that it's close as possible. Maybe not as close as, but it may still be moved a little bit to the right, I think, or maybe not. I mean, it's closest as possible to ABD. After we have the superimposition, we compute here, this is this similarity matrix, we compute based on the pairwise distances here, we compute this one, and we run structural alignment, the dynamic programming again, and after that we see that alpha, beta, gamma is matched nicely with ABE, and that will, probably after this position, their similarity score between them in our, when we compute this matrix again the next time, we will have a strong match scores between A, B, and E, and mismatch scores between alpha and C, alpha and D, beta and C, beta and D. They will have mismatch penalties because, as you can see, the distance between alpha and A, beta, and B, they are very small, which means that they will have high scores. When we run the dynamic programming again, it will probably return us the same set of matches. And at that point, it will converge. So that's the idea. Here's another figure from the original paper, which again tries to align A, B, C, D, E with this capital A, B, C, D, E, F, G. Different number of amino acids in both of them. So here the initial equivalences is A matched with C, B with this B. So this is the initial equivalence. If you try to superimpose these two with respect to this initial matching, this is the best superimposition you can get. Okay, if you, for example, rotate it a little bit, the distances between matched atoms is going to increase. So this is the smallest minimum RMSD you can get considering these matches. And after that, we compute a similarity score value based on the distances, and we run dynamic programming again, which changed our next matches. After these matches, we rerun the superimposition. Now the superimposition became this one. After the superimposition, distances have changed. We run the dynamic programming again with the new distances, and finally we converge to a new matching between these, where AB is CDE is matched with ABEFG, the capital ones, with an RMST of 0.23. So in 3 iterations, it converged. So this one just shows how it will improve at every iteration. This is the expected thing of this, but starting from a random alignment. But since this initial random matching is really important, it will give us our first score matrix. This is therefore the initial random alignment determines what we're going to get in the end. That's one of the biggest disadvantages of this method. All right, so that's it for the structural alignment methods that I want to discuss. Yeah, actually, since we have only 10 minutes left, instead of starting with the entirely new topic, I can stop here for today. Yeah, let me ask you one question before I go as an exercise. How do you think a parallel beta sheet, a set of beta strands which are parallel, a parallel beta sheet is going to appear in such an intramolecular distance matrix? You may write in the chat box. If I have in some part of my structure, if I have a parallel beta sheet, how is it going to appear in this intramolecular distance matrix? What do you think? Any answers? So I'm waiting, since writing a long answer may take time. So I will wait like for a minute or so. And meanwhile, if you, yeah, exactly. So both answers are good. It's in the direction parallel to the diagonal. So this is the diagonal, which is 0 with a slope of 1. So yeah, it will appear as a line segment here off diagonal. It will not be close to diagonal because beta strands are between two parts of structures that are far away in sequence, potentially far away in sequence, right? So it may be, if, for example, I may have, look at this region here, it's like, it's parallel to the diagonal. Maybe the relationship between these two parts may be about, part of it may be parallel beta strand. So they will appear as off diagonal. Off diagonal means they're far away from the diagonal, but parallel to the diagonal type of looking black regions. A black line with a positive slope is the perfect answer. All right. So if you have any questions from the structural algorithm or structural alignment in general, I will be happy to answer them. But this concludes our discussion on protein structures, and on Thursday we are going to continue with, so here's where we are on schedule. We are currently in week 11, and we will start microarray data analysis. I will briefly mention what microarrays are. Microarrays now are considered like an old technology these days, with these new techniques like RNA-seq, next generation sequencing. It's not microarray data, but transcriptomics data, so expression data, which tells us what is the messenger RNA quantity in the cell at a particular state. We are going to take a look at these, and I will talk about some problems to help us analyze the differentially expressed genes between different states, which may be between normal and people, or with the people with disease, between cancer and normal cells. So we will talk about clustering techniques, and we will see two algorithms, k-means and self-organizing maps here. It will probably I will not be able to finish all these in 1 hour, so we will jump into week 12 probably, but we still have 3 weeks left after that, only for biological networks. Probably I will spend the entire next week again with transcriptomics, the differential expression. Maybe we may talk about some differential expression methods too. And I will have 2 weeks to talk about biological networks. Okay, so that's the plan. Let me stop recording now.